if you lose integrity of your epigenome, so you will get epimutations, so you will get random alterations in your epigenome, you lose the template, not?
Welcome, everybody. This is Buck Joffrey with Longevity Roadmap, and today got a really interesting show for you. But before I start that, I just wanted to, I should, but I never ask people to, like the show, write a review, subscribe to the show. All of these things really help. The show is growing very quickly. We actually had 19,000 views last month, which is great for a pretty young show, and that's something I'd like to continue to grow. As you know, this is a very unique show where we're really just talking to real scientists and physicians and stuff like that, giving you the best possible, real information available out there, differing opinions often, but that's what we need to know. Anyway, if you could make sure you like, subscribe, write a review, whatever, and that'd be really helpful. Now, today, this conversation I have, I think will be very useful for many of you, which with Professor Jan Vijg. And he's chair of genetics at Albert Einstein College of Medicine, really smart guy who is really one of the pioneers looking at mutations, at point mutations, mutations in, genes, and then epigenetic drift, these terms that we hear about all the time when it comes to aging. So it's a real opportunity to really get a deep dive explanation on exactly what those things are. Why are they important? Why are mutations in, regular cells, which we call somatic cells, why are they important? Why and what exactly is epigenetic drift, and why is it important? We also talk about the concept of potentially reversal of epigenetic drift. He has interesting takes on that are different from others in the space. We also, I think it's notable that he mentioned a very interesting discovery, a molecule which is called the dream complex, which seems to be showing up in like germlines, in other words, like sperm and egg, where you don't get mutations in those, right? Because if you did, we would never survive from one generation to another. That molecule also seems to be more common in sharks, for example, where those sharks never get cancer and live forever, right? So anyway, really fascinating conversation. If you're into this stuff, you want to learn more about, what exactly is a mutation? Why is it happening? What is epigenetic drift? You're going to get all that information in this interview, and we will have that after these messages. Hey, longevity enthusiasts, it's time to take it to the next level. I've been fine-tuning my longevity regimen for years, and I look better and feel better than I did a decade ago. In fact, my blood work is even better than it was back then. And it's all because of my data-driven regimen, and it's inspired me to create a course and community just for you. It's called the Longevity Roadmap, and I urge you to check it out. If you're tired of your belly fat, tired of being tired, or just want to optimize yourself for the next 50 years, visit longevityroadmap.com. That's longevityroadmap.com. Welcome back to the show today. Today, my guest is Professor Jan Vijg. He's a chair of genetics at Albert Einstein College of Medicine. He earned his PhD in biology from the University of Leiden in the Netherlands, where he trained as a molecular biologist and went on to become one of the world's leading researchers on aging. Professor Vijg is a pioneer in the use of transgenic models to measure DNA mutations in living tissues, and now his lab applies single-cell genomics to study both genetic mutations and epigenetic drift. Welcome to the show, Dr. Vijg.
Sure, it's a pleasure.
Let's start with some, just getting some of the foundation out of the way, just so that people understand. People often use the word mutation broadly. Perhaps you could clarify the difference between permanent changes in DNA sequence, actual gene mutations, as opposed to what we call epigenetic drift.
Yeah. Well, epigenetic drift, has actually nothing to do with the basic structure of DNA, it's modifications of DNA. For example, a very frequent type of epigenetic modification is methylation, when one of the bases, a cytosine, is actually methylated. There's a small methyl group is hanging on there. And you can take that off and you can put it on. And those are functioning as signals for whether a gene, for example, is active or not active. When it is heavily methylated, as we say, so there are a lot of epigenetic components there on the gene, then it's usually silent. And when you can take it off by whatever mechanism, then the gene is active. Now, we call changes in those molecules, in those modifications, we call them actually epimutations. So that came later than the concept of mutations. Mutations are known since around 1900, when we didn't even know the structure of DNA. So we couldn't know, of course, that mutations were alterations in the DNA sequence because we didn't know DNA. But people did know that those were heritable changes. For example, they could find out that in flies or also in mice, that the coat color was determined by a particular gene. And if you inactivated that gene, and they didn't know, of course, how that worked, but they could see when they irradiated these flies or these mice, they saw a change in coat color. And they called that mutations, heritable changes. And it was only later that we realized that those mutations were actually changes in the sequence of the DNA in particular cells, skin cells or cells wherever in your body.
Yeah. So, I mean, just for the audience, just to sum that up, essentially mutations are in the DNA, and the drift that we're talking about are changes in chemical changes to the DNA that will change fundamentally sometimes how that DNA is read. And so it can basically turn genes on and off because of those kinds of changes.
Yeah,
is that a fair? Yeah,
is fair. And you can also say that epigenetic changes, what they call epimutations, are actually quite frequent. They're much more frequent, maybe 3 orders of magnitude more frequent than actually changes in the sequence of DNA. And that reflects the relative importance of that. Because when you really have a mutation, a permanent change in your DNA sequence, that's much more serious than when you change one particular base, a cytosine, you take the methylation group off. It depends a little bit of where it is, of course, but in general, the effect of that is much, much smaller than an actual DNA sequence mutation.
Right. So let's talk a little bit about the DNA sequence mutations in aging. What kinds of mutations do we see in particular that seem to relate with aging or accumulate with aging?
Yeah. Well, I mean, you raise an important point because there are many different types of mutations. The most frequent type of mutations is simply when you replace one base pair, 2 bases that are complementary to each other, so like a G to C, and you replace that for another one, for example, an AT. That's a very small change. And most of these changes have, as far as we know, no effect. Some can have an effect. Now, a more important type of mutation is the loss of an entire base pair. You really lose it, that base pair. The frequency of such changes has to do with the seriousness of it. When you look at this one base pair loss, that is tenfold less frequent than a replacement of a base for another base. And that has to do, obviously, with the fact that these occasional base replacements, we call it base substitutions, quite often there's no effect at all. So the genome can allow many of these mutations to occur because their effect is not that dramatic. But when you lose a base, that's more important. So it cannot afford to have too many of those. And especially when you lose large chunks of DNA, like whole chromosomes or chromosomal chunks, large pieces of DNA, those are very rare because, again, if they happen, the effect of that is almost always very large. By the way, what I didn't say, you didn't ask it, but how do mutations arise? Because interestingly enough, the whole idea of how they happened came later than the concept of mutations, because Watson and Crick published the first paper where they described the structure of DNA in 1957 or so, I believe, or 1954. I can't exactly remember, but right early 1950s, let's say. And then physicists Leo Szilard and Gino Faya, they actually published papers in Science and in the New York Academy of Sciences, hypothesizing that aging must be due to the accumulation of mutations, because they said those are probably causing cancer as well. And we now know, of course, that is exactly right. And cancer is an age-related disease. If ever there was one, that's cancer. Cancer is only really occurring, well, there are cancers of children, but they're very rare. It's really older people who almost always get cancer. So their argument was mutations accumulate, and therefore your chances of getting cancer increased with age.
Right.
That was the whole argument here.
And it's pretty much, for the most part, can we say that the mutations that we get as we age are largely environmental, or would you say that?
That's also a very good point. Some of them, of course, can be environmental, because people have heard of people exposed to radioactivity. Think of the nuclear bomb in Hiroshima and Nagasaki. And people, of course, who were not killed immediately but lived a little bit further away, got cancer. They got cancer because there's a radiation, and radiation can induce mutations. It's a very, very well-known mutagen. There are also chemical mutagens. However, under normal conditions, people are not exposing themselves to this kind of radiation or mutagenic chemicals. And then we have endogenous, we call them endogenous factors. And for example, think of free radicals. Free radicals are byproducts of breathing or using oxygen.
Living. Yeah.
Pretty useful process, not? Yes, but there's a very tiny amount of side products, and those we call free radicals, activated oxygen. And they can damage DNA. That's very well known now. But there are also other molecules, normal metabolites, that can cause DNA damage. In fact, DNA damage is very frequent. There are probably 100,000 lesions in the DNA per cell per day. And when I say DNA damage, I'm not talking about mutations. DNA damage is a break in the DNA, or it is a particular chemical molecule that's attaching itself to the DNA, or it's a cross-link between two pieces of DNA or a protein in DNA. Now, that is very frequent, but we have fantastic DNA repair systems. They are continuously monitoring the DNA for this kind of damages, of which there are many. And when they repair it, they usually do it very well. Everything is reconstructed perfectly. But there are errors, and the errors are mutations. When they have to resynthesize a piece of DNA, for example, they cut it out because there's a damage. Then they resynthesize it, they make a mistake. They put the wrong base in. There you are. That is a mutation.
Yeah. So I guess one of the questions, because we want to kind of get to the root cause of aging in some way. And so we know we'll talk about epigenetic drift in a moment, but do these somatic mutations, do you think it's fair to say they are one of the causes of aging, or are they the result of aging?
Yeah, it's another excellent comment. I said I use that sentence in my own papers occasionally. It's are mutations a cause of aging, or are they simply a consequence? Obviously, we do not know that. There are reasons to assume that it is a causal factor. I just mentioned cancer, which is an age-related disease, and that the risk of cancer increases dramatically with age. And we also accumulate mutations with age. We know that. We found it. Others found it by now. So it's really not known. On the other hand, mutations are actually frequent, although are actually not so frequent. I'm sorry. I say that because initially we thought that mutations were 100-fold less frequent than they actually proved to be. This is why I had problems in getting my first papers published, because people said, well, everybody knows that mutations are not as frequent as you found them to be. And I said, well, but this is what we found, and what can you argue? Now, it turned out we did know a little bit about the frequency of mutations. How could we know it? Well, from germline mutations. We could know whether there were changes in the germline, because you can see those from parents to children. But it turns out, as we showed not even that long ago in a paper, that mutations in the germline are much less frequent than somatic mutations. So we have great systems to keep our germline clean. And you can imagine, of course, why that is. If you have a high mutation frequency in your germline, as a species, you will not survive. it's basically the species becomes extinct immediately. But somatic cells, you only need them for one lifetime, not? So basically, why would you need to keep the mutation frequency low in somatic cells? That we know, but that's actually another argument to say that mutations are probably functional and they could cause aging. Because again, nature in its wisdom didn't even try to give somatic cells a perfect repair. And that's why mutations, you said. But still, we don't know the answer to it. I cannot answer you that mutations are the cause of aging.
Can we, I'm sure scientists look at germlines and the repair mechanisms there that seem to be, nearly perfect. Otherwise we'd be, we wouldn't survive as a species.
Exactly. Yeah.
And try to figure out or understand, how we can apply some of the things that we know about the germline to try to combat somatic cell mutations.
Yeah. Together with a very good friend of mine in Germany, Bjorn Schumacher, he discovered in worms a complex that we know is the dream complex. It's an abbreviation, DREAM, whatever. It doesn't matter. It's called the dream complex. The dream complex was discovered because it basically was responsible for the fact that cells were quiescent. So they couldn't divide anymore. But in the germline, in germ cells, it's not expressed. That's what he found out. But he noticed that in somatic cells, it is becoming expressed, it's active, and then it suppresses DNA repair. Now you wonder, why is that? Well, when it's not active in the germline has a very good DNA repair, as it is said. Somatic cells do not need a very good DNA repair. So the evolution in its wisdom decided to temper down DNA repair because it is costly, energetically costly. So why would you spend all that energy on cells that are basically don't live, are not immortal, but basically last only a lifetime? But that gives us a handle on it, because we could try to deactivate DREAM in somatic tissues. And this is exactly the experiment that we are currently doing. We are working on that experiment now. We have done some first results only days ago, and they seem to suggest that indeed, when we lower DREAM activity in somatic cells, there's less of a mutation rate. Mutations are going down a little bit because the repair is better, apparently. We activate DNA repair. Turned out there's another research group, Trey Eidecker in University of California, San Diego, who worked on the same thing, and he found exactly the same results. We hope, of course, that that's true, and then we have a handle on improving DNA repair.
There's also another example where there's probably, there's virtually no cancer, for example, and that's in sharks or in other species. Do we find similar, this dream complex potentially involved there as well?
Well, yes. I just mentioned Trey Eidecker in this pre-publication of his. This is not peer-reviewed, so you can find it in bioarchives. He found evidence that that's indeed the case. Yeah, it's less active in long-lived species. Now, we just published a paper together with Vera Gorbunova. It will come out in Nature very soon because it was just accepted, and we just got the proofs of it, where she had cells of naked mole rat. She had cells of bowhead whale. She also had cells of naked mole rat. She has lots of species. But we're talking about bowhead whale. That paper is about bowhead whale. Bowhead whale is an enormous animal, as you know, and it lives very long. It lives probably like 230 years. And we looked at those cells at mutation rates, and they were indeed low. Lower than humans, much lower than the mouse. And Vera looked at the repair processes, and she found that the repair processes in cells from bowhead whale are really much higher than in human cells, other cells. So again, it looks like we slowly begin to understand the role of DNA repair, DNA damage, mutations in longevity. You may ask me about epigenetic drift. That, of course, we don't know yet, but that's probably also a consequence of DNA damage.
So, yeah, let's switch to epigenetic drift a little bit. When you track epigenetic drift across tissues, what patterns stand out in the sense that do some organs lose regulatory control faster than others?
Well, this is not something that people have studied extensively. we are trying to do that now, but it's, we can detect mutations, but to detect AP mutations is really not that simple. The first tools to detect mutations was our mouse model is a selective system, but our single cell analysis system, single cell sequencing, was sort of the first tool to do that. Now we have better systems, cheaper systems, you can do much more. But for AP mutations, we and others published papers also on single cells. And so that's how we found out that the frequency of AP mutations, so changes in the epigenome, is far higher than mutations. But these are very difficult techniques, and it's very difficult for us to find out which epigenetic trigger is loose in which tissue. If there are tissue specificities there, it's really not very well known.
Especially in the longevity space with biological clocks and all these things, they're looking at epigenetic drift and trying to give a biological age and that kind of thing. I'm just curious in terms of, again, sort of framing the question back to when I asked, what is causal versus, which causes which? Aging causes epigenetic drift, or epigenetic drift causes aging. A lot of people seem to think that epigenetic drift is one of the big keys to the aging process. I'm curious what your thoughts are there.
Well, ultimately, it could be important, but I think it's part of a more general phenomenon. Our cells, they are functioning because there is a network of genes that interact with each other and that also interact with what we call gene regulatory sequences, like promoters, but also enhancers. So the part of the genome in a typical differentiated cell that is important for this kind of control of gene expression is probably 10%. So it's not small at all. Now we have the idea that very gradually during aging, the cells lose control. They lose regulatory control. We sort of made a discovery at some point that cells in young, for example, we use a mouse model to study that, and we looked at the heart, different cardiomyocytes in the heart, and we looked at the expression level of multiple genes, and they were exactly the same from cell to cell. So we look at gene X, and it is expressed to the exact same level in one cell, in the other cell, in the next cell, within a young mouse. But if you looked at an old mouse, you could see that was no longer the case. The same gene was now expressed to different levels in different cells. We call that transcriptional noise. But that's essentially also what you are just saying. In a way, you talk about epigenetic drift, but the epigenome, of course, is controlling the expression level of genes, not? When there is something like epigenetic drift, you will probably get this kind of transcriptional noise, so that cells begin to deviate from a fixed level at which they are supposed to express a particular gene, and now instead they begin to fluctuate, so they lose control. Now that ultimately could be due to epigenetic drift, but epigenetic drift could ultimately be due to mutations. That's also, I just mentioned Ray Eidecker. He published a paper in Nature Aging, I believe a year ago or so, where he showed that one mutation can change an entire block of epimutation, of epigenetics, of methylation actually. So in other words, there's a relationship between all these different levels. So you can have random changes in the DNA sequence. They are linked to random changes in the epigenome, which are again linked to random changes in what we call the transcriptome. So all the transcriptionally active genes. And together that wreaks havoc on your whole regulatory system in your cells. So it's not that your cells all die and stop functioning, not at all. That's also not what aging is. Aging is not that. You don't drop dead instantaneously. You gradually degenerate. Well, that could be perfectly explained by this kind of loss of gene regulatory control. Think, for example, of a heat wave. There's a very old person, I remember the story in France, this is already a couple of years ago, got out of his car, walked, he had a beautiful house, so he had to walk for a couple of minutes to the door. He died because of the heat. He had heat. Okay, we all have very fancy systems that can absorb this kind of heat, not? There are beautiful thermostats. They're all combinations of gene activities. But of course, it still worked in this person also, not as good. It was degenerated. And that could perfectly well be explained by all these gene regulatory patterns that sort of gradually lose control. And that's probably what happens during aging as well. Think of the immune response, for example.
One of the things that people often talk about, again, related to biological clocks, is that some epigenetic changes are potentially reversible. Your comments on that, I'm curious about this sort of efforts by some with, various, types of things like fasting or various other methodology to sort of try to reset their epigenome. Is the epigenetic drift or these changes, are they reversible to a certain extent?
Well, it's a difficult question. I think most of them are not. And the reason is actually fairly simple, because if you lose integrity of your epigenome, so you will get epimutation, so you will get random alterations in your epigenome, you lose the template, not? So how will you be able to revert that and bring back the correct pattern based on what? There's no tape somewhere stored in your body that can tell you where all these methylations need to go, or these histone groups need to go. You don't know that. So I don't think that's possible. It's very similar to mutations. If there's a mutation, you lose a piece of DNA, or there's a base substitution, there's no template left. When there's a template left, yes, then you can do that. That's why DNA is double-stranded. So when you have DNA damage on one strand, your enzymatic systems use the other strand to put the correct base in again. But you cannot do that when you begin to randomly lose epigenome chunks. That is possible, of course, because think of DNA replication. When cells divide, they also have to break down the whole epigenome, and they have to build it up again. Now, we still do not know the details of that process, but we do know that methylation, for example, can be restored by particular enzymes that use the other strand of DNA, their sister strands, of course, and they can use the other strand then as templates. It's a very complicated process, but they do manage to do that, but not perfect. They make errors there. So it's only let's say 95% of the epigenome which is correctly restored. So it is also true for DNA repair of DNA damage. If you have breaks in DNA or other chemical damage, enzymes are coming in and they take away the epigenome. They have to repair the chemical damage to the DNA, and then when they do that, they have to put back in the correct epigenome. Now, they can do that. They also have ways to do that, but again, not perfect. So that's why I do not think that you can really perfectly, to some extent, you might be able to do that, because you can imagine epigenome can also be the same from cell to cell. It can be a program. You can imagine that there are epigenetic changes that are part of a program that's doing something to your cells. Like, for example, memory formation in the brain has been argued to be part of a program where you change the epigenome. But that kind of changes, because they are really consistent, they are the same in every cell, not random. You can do that. You can probably fix that problem. As soon as there's a sort of a pattern that you can store somewhere, you're able to do that. But then you have stochastic changes, as you call it, random changes, you cannot. So I think to reverse aging in that way will probably prove to be impossible. But again, whatever I say, I mean, I can only answer questions based on what I now know. But there are of course always things that I don't know, as you can imagine. So it could be that 10 years from now, or 20 years from now, or 100 years from now, I will be proved wrong. But for the moment, I think it's not possible.
Some groups out there are, have been reporting, experimenting with Yamanaka factors to reprogram cells and potentially reverse epigenetic drift and that kind of thing. Tell me about what your thoughts are on that.
Yeah, I think that's absolutely fascinating, because first of all, we now know that using the Yamanaka factors, you can bring a cell back from its differentiated state into a stem cell state. Now, there's also something they call that partial reprogramming. So what you do then is you do not go through the whole process. Nobody really knows how far you need to go or why this has beneficial effect. But it has beneficial effects on they did it, show that in mouse model. Not completely convincing in my opinion, that we really need to do a lot more research. But maybe when you have a little bit of an imagination, you can argue, well, there is actually evidence for a clear beneficial effect. In particular, mouse models that actually are not during normal aging, as far as I know, there's only one paper, but not even peer-reviewed. That seems to suggest that you can live longer in that way. But we do not really know what happens there. Is partial reprogramming, does it mean that some cells go through the whole process, but not all the cells? And then you have a few new cells, stem cells it is? Or is there something else happening? Now, we are doing experiments right now where we do reprogramming of somatic cells. So we get induced pluripotent stem cells, and we measure the mutation rate. And it seems pretty clear that the mutation rate in these stem cells is significantly lower than in the mother population of differentiated cells. So it looks, it's possible that what you do during reprogramming is you reprogram DNA repair. You make DNA repair from how it is in differentiated cells. You turn it more like a germ cell, a stem cell. A stem cell is closer to a germ cell than a differentiated cell. So we are now studying, of course, and I'm sure we're not the only one, if this reprogramming is doing exactly that, if it is improving DNA repair. If that is true, then you can also consider partial reprogramming without actually creating stem cells, but just the reprogramming process will have a beneficial effect on DNA repair. And if we can understand that better, then that would explain why partial reprogramming could be beneficial, namely because it improves your DNA repair, not? So that's the hypothetical, obviously, but it would, you see, it's always irritating when there seems to be a beneficial effect of something, you don't know why. It's not satisfactory to me. I need to know why that is. This seems like a logical explanation.
So from your perspective, big picture, and I always like to kind of ask people in your position this, is that if you had to, if you had to make a bet on something that materially could extend healthy lifespan, tackling mutations, the genome, correcting drift, where are you most excited about? Where do you see the most potential for those kind of large breakthroughs?
Well, in a sense, I'm not completely sure if what you mean with this question. I mean, there are two possible ways to interpret this question. One, you could say, and you sort of said that, we want to improve health span. I think there are lots of ways to improve health span, and we do it all the time. We've done it since the 19th century, and we seem to well on our way to continue that. There are lots of interesting molecules from metformin and rapamycin that do that. But there's no evidence that this kind of interventions are actually improving your maximum lifespan. Right. So are they changing the lifespan of Homo sapiens? Do they bring, in a previous paper in Nature, we sort of calculated that our maximum lifespan is approximately 115. So the question now is, can we improve that maximum lifespan to, let's say, 150 or 160? Now, to do that is a completely different question, you see, because now you're no longer asking for a health span, you're really asking for maximum lifespan. Can we basically move that barrier? Because as we argued in that paper, that 115 plus or minus whatever, a couple of years, is actually a barrier. It's a barrier, a biological barrier, and thus far we have not been able to go over it. We couldn't. So your question could also be, what do you expect to find in the future that take that barrier away? Now, that question is more difficult to answer, obviously, but that's sort of the research that we are doing. We feel that the topic of this whole meeting, like somatic mutations, epigenetic changes, are ultimately a consequence of damage to DNA. That we feel is a universal cause of aging. In one way or the other, either because it results in mutations, maybe because it interferes with healthy transcription, maybe it causes epigenetic drift, maybe it directly causes transcriptomic changes. We don't know all these things yet, but we are close. We're getting close now. So if we now find a way to intervene there at a very basic level, to bring back that whole ingenious system of preserving your genetic material longer by improving DNA repair, I think that would be the breakthrough. If we are able to do that, I think now we can slowly begin to think of really letting people live to 150, which of course opens up a can of worms. Not there are many other questions you can now ask.
Well, maybe you'll the dream complex.
The dream complex.
Maybe
exactly.
that'll be the answer.
Well, look, we always spoke. That's part of my own research.
Yeah. Thank you so much. I do appreciate your time, Professor, for being on the show today.
It was a great pleasure. All right.
Thanks for listening. A quick reminder that while I am, in fact, a surgeon, nothing I say should be construed as medical advice. Now, make sure to include your physician in any medical decisions you make. And also, if you're enjoying the show, please make sure to show your support with a like, share, or subscribe.